The stomach secretes hydrochloric acid at a pH between 1.0 and 2.0. That is a fluid corrosive enough to dissolve metal. Yet this highly destructive acid is housed inside an organ made entirely of delicate living tissues and cell membranes. How the stomach avoids digesting itself is a central problem of human physiology. We can solve this paradox by organizing the entire upper gastrointestinal tract into a three-part functional framework. If you are taking notes, color code these domains right now: propulsion in blue, secretion in red, and mucosal defense in green. This flowchart maps the domains onto the anatomy. Food is propelled down the esophagus. In the stomach, it undergoes chemical processing, while the wall maintains a mucosal barrier. This relies on mechanical digestion to crush food and chemical digestion to break bonds within macromolecules. When these systems face physiological stress, the tissues adapt. You must precisely distinguish between two terms. Hyperplasia is an increase in the absolute number of cells due to cell division. Hypertrophy is an increase in the physical size of the individual cells, with no change in the total cell count. If the environmental stress is chronic, tissues may undergo metaplasia. This is the reversible replacement of one fully differentiated cell type by another that is better equipped to survive the harsh conditions. This three-part framework provides a functional map for clinical diagnosis. In this model, a failure in propulsion results in disorders like achalasia or dysphagia. A failure in secretion leads to maldigestion. And a collapse of mucosal defense creates the conditions for peptic ulcer disease and reflux esophagitis. Let's look at propulsion. Swallowing, or deglutition, is an intricate neuromuscular reflex coordinated by the swallowing center in the medulla oblongata. It executes two simultaneous goals: driving the bolus forward and strictly protecting the airway. It begins with the voluntary oral phase. The tongue collects the chewed material, mixes it with saliva to form a cohesive bolus, and sweeps it backward against the hard palate into the oropharynx. This table and diagram outline the involuntary pharyngeal phase. The moment the bolus hits tactile receptors, the brainstem takes over. The soft palate elevates to seal the nasopharynx, preventing food from entering the nasal cavity. Simultaneously, the larynx pulls upward, causing the epiglottis to fold down while the vocal cords tightly close, sealing off the trachea. During this exact sequence, the medullary swallowing center overrides the respiratory center. This is deglutition apnea, a temporary mandatory halt in breathing that guarantees you cannot inhale while food is crossing your airway. With the airway secured, the upper esophageal sphincter relaxes. The pharyngeal constrictor muscles contract in rapid sequence, forcing the bolus through the open sphincter and into the esophagus. Once inside the esophagus, transit is governed by the law of the intestines, driven by the enteric nervous system. Mechanoreceptors detect the stretching of the esophageal wall. They trigger ascending interneurons to release acetylcholine, contracting the circular muscle behind the bolus. At the same time, descending interneurons release nitric oxide and vasoactive intestinal peptide, relaxing the muscle immediately ahead of it. This sweeping wave initiated by the swallow is primary peristalsis. If any food gets left behind, the local distension automatically triggers secondary peristalsis, which repeats until the esophagus is entirely clear. We see a catastrophic failure of this system in achalasia. Pathologically, achalasia is the selective destruction of the inhibitory ganglionic cells in the myenteric plexus. Without the neurons that release nitric oxide and VIP, the lower esophageal sphincter cannot relax. Cholinergic tone dominates, leaving the sphincter permanently clamped shut. Once the bolus clears the esophagus, chemical processing begins. The engine of this processing is the parietal cell, which pumps hydrogen ions into the gastric lumen in exchange for potassium via the apical H+K+ ATPase pump. This diagram illustrates the receptor-level integration at the parietal cell. The output of the acid pump is driven by three synergistic stimulators: acetylcholine from the vagus nerve, gastrin from antral G cells, and histamine from ECL cells. Histamine is a powerful paracrine signal. It binds to H2 receptors on the parietal cell, increasing intracellular cyclic AMP. Note the receptor type carefully. These are H2 receptors governing gastric acid, distinctly different from the H1 receptors involved in allergic responses. Gastrin amplifies this process through a dual mechanism. It binds directly to CCKB receptors on the parietal cell to raise intracellular calcium, but it also binds to CCKB receptors on nearby ECL cells, triggering a massive secondary release of histamine. Left unchecked, this would acidify the stomach endlessly. The master physiological brake is somatostatin, secreted by E cells. When the luminal pH drops below 2.0, somatostatin is released, directly inhibiting the parietal cells, the G cells, and the ECL cells. There is also an intestinal feedback loop. When nutrient-rich acidic chyme enters the duodenum, enteroendocrine cells release secretin and gastric inhibitory peptide, or GIP. These hormones suppress parietal cell acid production and slow gastric motility, giving the small intestine time to neutralize the acid. When regulation fails, the results are severe. In Zollinger-Ellison Syndrome, a neuroendocrine tumor called a gastrinoma secretes autonomous, continuous gastrin. This overrides the somatostatin brake entirely. The resulting acid hypersecretion overwhelms the duodenum, causing intractable ulcers, while the massive gastrin levels drive trophic hypertrophy of the gastric mucosa. This brings us back to our initial paradox. To break down proteins and kill pathogens, the stomach intentionally maintains a highly destructive environment. It survives this by isolating the acid from its own tissues. This cross-section shows the architecture of the gastric mucosal barrier. It begins with physical isolation. The epithelial cells secrete a thick, continuous mucus gel layer. Beneath that, high-resistance tight junctions weld the epithelial cells together, strictly preventing hydrogen ions from leaking back into the submucosa. Trapped within the base of that mucus gel is a high concentration of bicarbonate ions. This creates a sharp, localized pH gradient. The fluid in the central lumen might be pH 1.5, but right at the surface of the cell membrane, the trapped bicarbonate neutralizes the acid to a safe, neutral pH of 7.0. Beneath the epithelial layer lies a dense microvascular network. This mucosal blood flow is an active defensive mechanism. It supplies the oxygen and bicarbonate needed for cell repair, and it rapidly flushes away any stray hydrogen ions that manage to breach the tight junctions. The master regulators orchestrating this entire defense grid are endogenous prostaglandins, specifically PGE2 and PGI2. They continuously drive the secretion of mucus, pump out bicarbonate, and dilate the mucosal blood vessels. This balance model explains peptic ulcer disease. Ulceration occurs when offensive factors outstrip the defensive factors, allowing acid to burn through the muscularis mucosae. This flowchart tracks the two primary drivers of that collapse. The first is nonsteroidal anti-inflammatory drugs, or NSAIDs. Drugs like aspirin and ibuprofen systemically inhibit the COX-1 enzyme. This completely shuts off prostaglandin synthesis. Mucus drops, bicarbonate drops, blood flow constricts, and the defense barrier collapses, resulting in silent, deep ulcers. The second major driver is Helicobacter pylori. This bacteria colonizes the mucus layer and produces the enzyme urease, which converts urea into a protective cloud of ammonia. The ammonia buffers the acid, allowing the bacteria to survive, but it also destroys the mucus gel, breaks the tight junctions, and triggers chronic inflammation that erodes the tissue. Additional environmental factors exacerbate the damage. High concentrations of ethanol directly dissolve the lipid membranes of the epithelial cells. Nicotine from cigarette smoking restricts mucosal blood flow, severely delaying the tissue's ability to heal. Peptic ulcer disease is rarely a problem of producing too much acid. Aside from gastrinomas, it is almost exclusively the systemic collapse of the barrier mechanisms that safely contain normal physiological acid levels. To consolidate this material, look at how the three pillars dictate the system. The necessary forward transit of food requires intense chemical processing, and that processing inherently mandates an impervious physical barrier. For airway defenses, remember the three involuntary pharyngeal safeguards. The soft palate seals the nose, the epiglottis and vocal cords seal the trachea, and deglutition apnea suspends the respiratory drive. Do not fall for receptor pharmacology traps. Achalasia is a failure of relaxation due to an absence of nitric oxide and VIP. And when dealing with acid suppression, parietal cells are stimulated by H2 receptors, completely separate from H1 allergy pathways. In tissue pathology, keep your terms exact. Hyperplasia increases cell number. Hypertrophy increases cell size. For high-yield multiple-choice questions, H. pylori survives gastric acid specifically by secreting urease, and the master inhibitory brake for acid secretion is somatostatin, released exclusively by D cells. If you were asked to explain hematemesis, vomiting blood, in a patient taking chronic ibuprofen, trace the pathway. The NSAID inhibits COX-1, which depletes prostaglandins, which halts mucus and bicarbonate production, allowing normal acid to erode into a blood vessel. Mucosal blood flow acts as a dynamic, active defensive mechanism. The subepithelial capillary network delivers essential oxygen and bicarbonate to the surface cells, while simultaneously flushing back diffused hydrogen ions into the systemic circulation. Master the tripod. The upper GI tract must propel the bolus without compromising the airway, chemically process macromolecules without destroying the host, and actively maintain a multi-layered shield to survive its own environment.